Hello friends, this video on molecular basis of inheritance part 14 is brought to you by examfear.com. No more fear from exam. So let us now talk about the machinery and the enzymes. Now when we say machinery, what do we mean is, as I said, the process of DNA replication. Okay, so for this process to take place, it is not only DNA that is required. There are quite a couple of additional stuffs which are also required to make the process of replication possible. And one important thing is the enzymes. Now, we all know what are enzymes. Enzymes, structurally, they are all proteins and they actually help to catalyze the chemical reactions. So, they act as catalysts. So, enzymes play a very important role in replication, acting as catalysts, because even during the process of replication, when we will look at the process step by step, you will get to know that why do we need catalysts for creating a copy of DNA. Now, some of the important enzymes which play a very important role during DNA replication is DNA polymerase. This is the most important enzyme which is needed for DNA replication. Like in any movie, you have somebody playing the lead role and then you have all the side actors, right? So in a very similar way, DNA polymerase here plays the main role. So the most important function is being performed by DNA polymerase. Helicase, primase, DNA ligase, these are also the enzymes which play roles during the process of DNA replication. So now we will have a look at the roles of each of these enzymes, and also we will see how the process of replication actually take place. So first we will talk about DNA polymerase and helicase. So what do they do? As I said, DNA polymerase, they create the DNA from nucleotides. So that is the main purpose, right? In the process of replication, what happens is, when I say they create DNA, that means it actually synthesizes the new strand of DNA from nucleotides. That is, the existing nucleotides will already be there, right? The template strand. For example, these two strands, they are the original strand, or this is the parental DNA. Now, the strands will separate out. Now, once the strands separate out, what will happen? These two strands will act as the template. They will act as the original copy. Now, based on the sequences on each of these strands, the new sequences will be created, and those new sequences will be joined together to form the new strand of DNA. So this entire creation of the new DNA is being done by the DNA polymerase. So that means the main role is being performed by DNA polymerase. So it will read the existing DNA strands, it will read the strand which is acting as a template to create the new strands. And then the new strand will be complementary to the existing strand. So DNA polymerase will be needed every time a cell divides, so that one copy of DNA can be passed to each daughter cell. That is the time when a cell should divide, because when a cell is dividing, it needs a copy of DNA, because the new cell will need a copy of DNA. So that means the replication process should take place before cell division. So every time cell division takes place, DNA polymerase is a must. So the strands will divide. Now here you can see these two strands are the template strand. So this enzyme will read the sequence on the existing strands, and based on that it will create the new strands with the complementary sequences. So basically this is the enzyme which will actually create the DNA copy. And finally it will give rise to a copy of DNA. Helicase is the enzyme which unwinds DNA from tightly double-stranded structure. So here you see this is the double-stranded structure. So from this double-stranded structure to two separate strands. So how does this separation happens? The DNA polymerase doesn't play any role during separation. So before DNA polymerase can do its job, the two strands need to be separated. And that separation is done by this enzyme called helicase. Why is it known as helicase? From the word helix or helical. So the helical structure is being unwinded, and that is why the enzyme is helicase. There is another enzyme, that is primase, and what does it do? Let us see. So this enzyme will create a short fragment of RNA, which is also called primer, paired with the template DNA strand. Okay, so I think you will not be able to understand the exact function of primaries right here, but as we move on to the next slides, you will get to know what exactly it does. So basically what it will do is, in certain cases, the DNA polymerase directly cannot create a copy of DNA. I will tell you why. Now, because here there are two strands which are obtained, which act as template. So, in one of these strands, DNA polymerase can independently create the complementary strand. But in another strand, it is difficult for DNA polymerase to initiate the process. So, somebody else has to initiate the process, and then DNA polymerase can do its job. Now, why? That will be answered a little later. So, for now, you just understand it like this, that DNA polymerase cannot initiate the process or cannot initiate its own function. So it needs somebody else to initiate the process. And who is that somebody else? That somebody else is the enzyme primase. So primase will just start the process. It will just, like if you have a function in your school, somebody is there to inaugurate the function. As long as the function, somebody doesn't inaugurate the function, I mean the events doesn't start off, right? So suppose if there is a play which has to happen, there is say a dance program which has to happen, until and unless inauguration is done, the programs won't follow. Similarly here, so primase will just initiate the process, and after that it will all be taken up by DNA polymerase. So primase, what does it do? It just creates a very short fragment of RNA, and after that DNA polymerase will create the bases, and then it will create the DNA strand. So it is called primase because the small fragment of RNA which is created by it is known as primer. So primer would mean something in the beginning. Primary, what is primary? Something which happens in the beginning or at an early stage. So that is why the name of the enzyme is primase. So I gave you a rough idea about what these three enzymes do. Now when we talk about the process, you will get to understand their role even better. Now, if you talk about DNA polymerase, it is an absolutely efficient and it does its function with absolute accuracy. So I would not say absolute accuracy, but yes, to a very high degree of accuracy. So it catalyzes polymerization of large number of nucleotides in a very short time, and that is how we see that it is very efficient, because it is able to perform its task in a very short time. So when we say that it is very efficient, that means it does its job in a very short time. What is its job? Its job is to polymerize the nucleotides, that is to create more base pairs. So how many base pairs does it create per second? So that would be known as the rate of polymerization or the rate of replication. Because polymerization is what? The process of polymerization means combining many monomer units to form a polymer. That is polymerization. And what is replication? Replication is creating a copy of DNA. So when polymerization is being done, that means a new strand of DNA is being created by joining many nucleotides. So when you are doing a polymerization, a replication is obviously done. So polymerization and replication would actually mean the same thing here in this context. So if you talk about the speed of polymerization of DNA polymerase, it has been found that in E. coli, one complete replication is done in 18 minutes. So in 18 minutes, one complete replication is done. So generation 1 is being created in 18 minutes. And there are how many total base pairs in an E. coli? So in E. coli, there are 4.6 into 10 to the power 6 base pairs that exist. So it has been found that if you try to calculate the rate of replication, it comes around 2000 to 3000 per second. So that means such a high number of base pairs, 2000 to 3000 base pairs are being created in just 1 second. So it is extremely fast. So the rate of replication or the rate of polymerization is extremely high. It also catalyzes with high degree of accuracy. So when we talk about accuracy, we mean that since it is all about replication, so we are basically creating a copy. So we should copy it in the right way. For example, if the original copy has a sequence somewhat like this, So if the original copy has this sequence, so we expect that the new copy which is being created will also have the exact sequence. But if somewhere by mistake, now mistakes can happen, it is not that it is error free. So mistakes can happen even by DNA polymerase, but at least it has got high degree of accuracy. A mistake is made in every billions base pairs copied. So that is very small, right? Just one mistake in 1 billion of base pairs that are being copied. However, DNA polymerase also have an option to proofread to check for the errors, and once it finds errors, then it can also rectify it. But in case these errors remain, if they are not rectified or if they are not caught during proofreading, if these errors remain, they give rise to mutations. So here you can see, if you look at this, let us suppose this was the parental copy of DNA and this is the new copy that is being formed. So you see there is a difference here. However, this polymerase will have the provision for proofreading. So we can see that DNA polymerase is a quite efficient and accurate enzyme. Thank you. Please visit www.examfear.com to watch more videos, attempt free online test, get free study material, find tutors and mentors. Thank you once again.